All right, so this lecture is going to cover cell signaling. We've already discussed nervous system, and you can see how the neurons basically signal or communicate to other cells. Um, Nervous system is one of those big systems that talk or signal to other cells, but there are multiple other ways. We're going to see endocrine system, but we're going to look at how the cells actually communicate. What is the communication taking place? And think of a human when they communicate, they have one person talks, right? The other person has the ears to listen, and we get communication. That's essentially what's taking place when we do cell signaling, where cells are communicating to each other. Cells can talk to cells locally to them, right next to them. Okay, so we'll see these different types of signaling and how they take place. There's two kind of main ways that cells communicate. They can communicate via gap junctions, and gap junctions are actually basically almost tethered together with these proteins, these gap junctions that are basically little tunnels from cell to cell. Okay, and you can see any ions moving through here can actually move through to the other cell. Okay, so that's a gap junction. We saw in the nervous system called an electrical synapse when we're talking nervous system. Um, but again, there is a direct connection between the two cells. We're going to see this when we look at the cardiovascular system, look at the heart cells. Okay. The other mode of signaling is chemical signaling, right? And in this mode, one cell secretes a chemical, basically is talking, saying the words. The chemical messenger can be the words, and the chemical messenger is going to be bound to the receiving cell or the listening cell. And you can think of the receptor as the ears. The ears are going to receive the words, and we're going to get communication from one cell to the other. Okay, and so most of our focus here is going to be on chemical messengering, different types of messages between the cells using a chemical as the messaging system. So some of the classes of chemical messengers, basically it's what cells are doing the talking and who they are talking to. One of those classes is a paracrine messenger. Okay, paracrine is a local type signaling, right? Where one cell secretes a chemical into the interstitial fluid, right? And the other cell has a receptor for that, and the cell can receive the signal. So you can see when we're doing chemical messaging, there's basically got to be two main components. We got to have a chemical messenger, but the cell that's listening has to have a receptor that binds that chemical messenger. Otherwise, we're not going to get a signal to the other cell. Okay, in paracrine again, it is a local. It is very local. It is not over long distances. It is from cells are talking to the cells just neighboring them. One mode of kind of long distance signaling is when we're using a neurotransmitter, right? These neurons, this neuron can be way up in the spinal cord, and this synapse can be all the way down at the toe. This could be a 3-foot long axon. But at the end of these axons is when we've seen in the nervous system, we have neurotransmitters. Neurotransmitters are the chemical signals from the neurons. The neuron secretes them, okay, exocytose into the synapse, and then the target cell will have to have a receptor. Again, to get chemical signaling, we need a chemical messenger, and we need the receptor on the target cell to respond to that messenger, to listen to the signal that's taking place. Okay? And again, neurotransmitter are released by the neurons, and the receiving cell, it can be many different types of cells that are the target cells of these neurons. Okay? Another class of chemical messengers. Again, this will be long distance signaling, not like the paracrine where it's very local. This is another form of long distance signaling because the signaling molecules will actually be secreted into the bloodstream. Okay? In our endocrine system, we have hormones that are going to be released by the endocrine cells, and the chemical messenger actually is picked up into the blood supply. So basically any cells that have access to the blood supply could be the target cells, could be the ones responding to the chemical messenger. But again, the target cell has to have a receptor for that hormone to be able to listen. Okay, you can see a non-target cell over here. There's no receptor for this hormone. This cell's not going to respond to it. But the target cell here has a receptor for this hormone, and you will get a response in that cell. Alright, so we have those class of chemical messengers. We've done the cells that are doing the speaking have talked with the chemical messengers, either the neurotransmitters, the hormones, or the paracrine chemical messengers. Okay, and remember, for chemical signaling to take place, we have to have the target cell has to have a receptor that will bind the chemical messenger or respond to it. Okay, so now we're going to look at the different classes or different types of receptors to get a response in the cell. So basically that chemical messenger is there to cause a change or response in the target cell that has the receptor. Okay, one class of receptors is we can have ligand-gated ion channels. Okay, ligand-gated meaning a chemical. In this case, it would be the chemical messenger. The chemical messenger binds to the receptor, and that opens up an ion channel. Okay, so here, channel is closed. There's no chemical messenger bound to the receptor. The channel is closed. No ions are moving through the channel. Okay, now the chemical messenger is around, and it binds to the receptor, and that will open up the ion channel. The ion channels can be various. It could allow for sodium, allow for potassium, allow for chloride, or some of them allow for both sodium and potassium, depending on what it is. What that can do is we can get a change in electrical property. Okay, we can get a change in the membrane potential, depending on what ion is moving through here. Okay, similar to what we've seen in the nervous system, we get changes in membrane potential with movement of ions with their charges going through there. Okay, another mode, a ligand ion channel, we can have calcium channels. Okay, again, it is a ligand-gated ion channel, but this case it is a channel for calcium ions. Okay, besides here, where we just got a change in electrical potential, calcium channels can have multiple effects. Okay, channel is closed. There's no chemical messenger. When the chemical messenger is available, it can bind to the ligand-gated ion channel and open the channel. Now calcium, because calcium is high outside and low inside, calcium will rush in. Okay, we can get multiple changes here. Calcium can change the electrical contraction properties in, say, muscles. Okay, it can cause secretion of various molecules, and we can also get this response where we get second messenger system moving, and we're going to be talking about second messengers, but one of those second messengers can be calmodulin. This gives you an amplified effect in the cell. Okay, and this can go change activities of various proteins. Okay, depending on what cells depends. So basically, I don't need you to know what actually the responses are, but the calmodulin can activate proteins, and that can alter activity in the cell. Okay, so first class ligand-gated ion channels, we're going to allow binding of the chemical messengers, allows various ions in. Depending on the receptor, depends on what ions are allowed through. Okay, if we're dealing with sodium, potassium, or chloride, that's going to change electrical property. The ion channels for calcium, we can get a much broader or different effects in the cells. Another type of receptor, enzyme link receptors. One of the common is called a tyrosine kinase receptor. Okay, in this case, there is a kinase enzyme that gets activated when the chemical messenger binds to the receptor. Okay, if you remember kinases are enzymes that can phosphorylate. Okay, and we can turn on and off proteins depending on whether they're phosphorylated or not. So if we are able to activate an enzyme by binding the chemical messenger, we can change activity by phosphorylating different proteins. Okay, depending on the cell, depending on the protein that gets phosphorylated, depends on what reaction takes place. But what we're doing is we're getting a change once the chemical messenger is bound. Okay, so just walking through the steps here, we have the tyrosine kinase receptor. It's inactive. The chemical messenger isn't bound. All right. Once the chemical messenger binds, that turns on the enzyme here, the tyrosine kinase enzyme. So going back to our enzyme days, what's a kinase do? It phosphorylates, it adds a phosphate group, and in this case it's going to add a phosphate to a tyrosine amino acid. So remember, amino acids make up proteins. There are various proteins with the tyrosine. You can see when the enzyme is active, it adds a phosphate group to it. Okay, and depending on what protein is, that could turn on a protein or turn off a protein, but by turning on or off these proteins, we can change or have a response in the cell. Okay, again, what's the focus or what's the function of the chemical messenger is to cause a response in the target cell. Okay, in this case, the response is going to happen due to either from phosphorylation of various proteins. So the broadest range of receptors out there are this class. They are the G protein coupled receptors, okay, GPCRs. All right, and basically what it is, it's binding of a chemical messenger are going to activate the G proteins that are coupled to the receptor. So looking at the receptor here, okay, here's the G protein coupled receptor. There's the receptor, and you can see these alpha, beta, and gamma are the G proteins. Okay, they are coupled to the receptor. Okay, when the receptor is not bound, these G proteins are coupled to the receptor. Okay, binding of the chemical messenger can cause activation of these G proteins. All right, and the G proteins, when activated, can go and cause responses in the cells. Okay, one response might be a G protein that is activated, gets uncoupled, and can open up ion channels, like we saw similar to the ion-gated channels. This is a slower response than the ligand-gated ion channels. Okay, but again, it would have a similar effect depending on what channel this is. If it's a sodium, a potassium, or chloride, it would cause changes in membrane potential. If it's a calcium, it could have various effects. It could change membrane potential, or it could cause calmodulin Okay, that second messenger system. Okay, so that's one effect. The G protein. So G proteins, besides opening ion channels, G proteins can also activate second messenger systems. Okay, one of those, the most common pathway or second messenger system, is the cyclic AMP. Okay, cyclic AMP is a second messenger system. All right, so let's walk through the steps. And here's our G protein couple receptor. All right, the messenger hasn't bound yet. The G proteins are coupled. Once we get binding of the messenger, now we get activation of these G proteins. They get uncoupled, and you can see instead of opening an ion channel, it is activating this enzyme that is in the membrane, adenylate cyclase. All right, what does this enzyme do? It takes ATP and converts ATP into cyclic AMP. Okay, cyclic AMP that can go on and activate various proteins, and we can get responses in the cell. What we're going to see with these G proteins, or sorry, the cyclic AMP, the second messenger system of cyclic AMP, is that what we're getting by binding one chemical messenger, we can actually get multiple G proteins activated that can activate multiple adenylate cyclase, and that multiple adenylate cyclase can convert many molecules of ATP into cyclic AMP. So what we're getting is from this one messenger, we're getting this amplification of the signal inside the cell. Okay, so let's take a look at what's taking place with this amplification. So binding of one messenger, we can get a broader effect in the cell by using this second messenger system. All right, so let's see how this amplification takes place in the cyclic AMP second messenger system. Okay, we got our G protein coupled receptor. It takes one chemical messenger molecule to activate the receptor, and that activation can then activate several G proteins. So we went from one chemical down to 10 activated G proteins, and each of those G proteins can then activate one adenylate cyclase enzyme. So we 1, 10, 10, and now that adenylate cyclase can basically convert many molecules of ATP into cyclic AMP. So we went from 1, 10, 10, now up to 5,000 molecules of cyclic AMP are created. Okay. Each of those cyclic AMP can then activate a kinase protein. Okay. And then each of those kinase proteins or kinase enzyme, it's an enzyme because it's going to phosphorylate molecules. We went from 5,000. Those protein kinase can do phosphorylate many molecules, many proteins. And now we went from one chemical messenger into 2.5 million phosphorylated proteins. So you can see how we get this broad response inside the cell. From one binding to one receptor, we get this amplification of the system in the cell, so we can get this larger response in the cell. Okay, so that is that for the cyclic AMP, the second messenger system. This is one of the more common ones, this, and we may see calmodulin, which we saw earlier, as second messenger signaling that's going to take place in the cell. Okay. After that, we got, so all the molecules so far, all the receptors we saw, those chemical messengers can be polar. They can be hydrophilic. They can be polar molecules. So because the receptors are here out on the membrane, they don't have to go through the plasma membrane. there are chemical messengers that are lipophilic, all right, nonpolar molecules. Many of your steroids, your hormones, your testosterone, progesterone, estrogen, they are made from cholesterol, which is a very nonpolar molecule, so they are lipophilic messengers. So lipophilic messengers, because they're small and lipophilic, they can diffuse directly through the membrane. Okay, therefore, the receptor doesn't have to be on the plasma membrane. So this is where we get intracellular receptors. Okay. And you can see for these lipophilic messengers, they can diffuse in. The receptor is not here out on the membrane, it is out on the, or in the cytoplasm. Okay, or it can be in the nucleus even. Okay, so cytoplasmic or nuclear receptor. Okay, but the response is almost the same in that these intracellular receptors, when bound, are going to bind to the DNA. They're going to bind to a promoter region where there is a certain gene to be transcribed. Okay, and therefore these intracellular receptors act as what we call transcription factors. Okay, so these receptors are going to cause for transcription and eventually will result in translation into proteins. So binding of that receptor results in certain proteins being produced. Okay, so remember that with our intracellular receptors. We're going to see these again. We'll talk about them a little more in depth in the hormone section when we start talking about testosterone, progesterone, and so forth. But realize these intracellular receptors can be inside the cytoplasm or in the nucleus, and ultimately they're going to result in transcription and the production of various proteins, depending on what gene, okay, the receptor binds to promote transcription. Depends on what protein is going to be produced. Okay. So that's it for cell signaling. So we got nervous system and cell signaling for this exam, and that is it.